thank you for getting up at this graveyard hour on the last day of ASMS. I hope I can excite you a little bit about proteomics now, switching gears from metabolomics. I'm sure here in the audience everyone knows what a chimeric spectrum is, and in DIA we actually generate them on purpose. However, as we've seen previously and others have shown before us, also DDA spectra can actually be substantially chimeric. And this is visualized here by looking at a library of 400,000 precursors, and we plot them along the MZ axis and the retention time axis. And we bin them in bins of 2 Thomson by 1 minute. And if you look at those bins, in the majority of all cases, you will actually pick not only a single precursor in DDA, but multiple. So in order to really tackle this problem, not in DIA data per se, but in DDA data, we've developed an AI-driven algorithm for the deconvolution of chimeric spectra. And what we're doing here is really we take all peptide candidates in the isolation window and we let them compete at the same time for the experimental intensity. We don't do spectrum duplication, we don't do subtractive methods, we don't remove peaks from the experimental spectrum. The experimental spectrum stays as it is. And these peptide candidates basically compete, and we then extract the contribution of each single peptide to the experimental total ion chromatogram. And if we do that for this particular spectrum here, for example, then we can identify not only 1, not only 2, but 3 and even 4 peptides in this particular experimental spectrum. And since I mentioned we are doing that AI-driven, we predict 2 things. One is the fragment ion intensities, and we predict retention times. And as you can see here, the fragment ion intensities on top of the x-axis, they correspond very well to the experimental fragment ion intensities below the x-axis. But if we now focus on the retention times, those suggest that those 4 peptides co-elude. And if we look in the isolation window at MS1 level for that particular MS2 scan, we see that all of these precursors are actually present. This particular algorithm is implemented in Chimeras. You might have seen a couple posters on ASMS using Chimeras already. And Chimeras is integrated in Proteome Discoverer 3.0 and really consists of 5 different steps. We start, as I mentioned, with the prediction of peptide properties. And then we do a first search that is not using retention time as an information. We generate a couple 10,000 PSMs and use those PSMs to recalibrate our AI to your data. So we want to make sure that we predict at the correct collision energy, and we want to make sure that our retention times are actually adapted to your chromatographic setup. And once that's done, we can do our main search with deconvolution and error estimation through percolator. And I'm not going to bore you with the results on DDA. This is a DIA talk, just suffice it to say. In DDA, it works very nice. We increase our PSMs by nearly 4-fold, peptides by nearly 2-fold, and add 50% more proteins, which I think is pretty encouraging results. And that is all in comparison to previous versions of Proteome Discoverer using sequest and variations of it. And what is very encouraging is that these previous versions, all these peptides that they were identifying, or the majority of them, we can identify with Chimeras and add more on top of that. But since we now can deconvolute chimeric spectra, one cool thing is that we can now adapt our measurement to the way we analyze our data. So we can now acquire data using wide windows, so wide window DDA experiments. And here I've plotted on the left-hand side basically the number of PSMs that we can obtain if we sort of ramp the isolation window from 0.4 Thomson all the way to 8 Thomson. And you see with Chimeras, those PSMs rapidly rise and then saturate as you would expect. But since once you open the isolation window, you have to make sure that all these additional peptide candidates that could match your experimental spectrum, that you don't get an overestimation of FDR because, of course, you increase your chance for random matches. So what we've done is entrapment experiments where we took a human FASTA file and concatenated it with a 9-fold shuffled version of it. So we shuffle peptides we shuffle peptide sequences at the peptide level, and then search that same file or 4 different files with that concatenated database, and then plot target decoy FDR on the x-axis here, and on the y-axis, we plot entrapment FDR based on the identifications into shuffled sequences. Since each shuffled sequence is per definition wrong, we can have that entrapment FDR. And if you look at the curves for sequenced HT with precursor detector, a algorithm dedicated for the analysis of chimeric spectra, you see that as you increase the isolation window, you get a bit more matches into the entrapment space and into the decoy space, meaning that we are a bit anti-conservative with precursor detector. However, for chimeras, that's actually not the case. So no matter the isolation window, whether it's 0.4 Thompson or 8 Thompson, our entrapment FDR looks very good. And just for those that are not used to looking at those plots, you want your curve to be on the diagonal, as close to the diagonal as possible, meaning that you get as many hits into your entrapment space as you get in the decoy space. So that begs the question, do we actually have to decide? I've shown you what is out there right now. You can use chimeras for DDA data. So where do we go from here? What do we do now? Do you actually have to take the red or the blue pill, or are they actually different from one another? So traditionally we've heard already that peptide ID and quantification and DIA is usually done a bit differently than in DDA. Normally, you do identification and quantification in parallel, you're looking at XICs, whereas in our spectrum-centric approach, we actually don't do that. We do ID on the currency that we all measure, the spectra, and then we do quantification using these contributions that I've mentioned earlier. On MS1, as currently implemented in Proteome Discoverer, that is done by Manora feature detector, but in MS2, we can use these contributions that I mentioned earlier, trace them across retention time, calculate the area under the curve, and quantify peptides that way. And you might think, well, DIA spectra and DDA spectra, they look completely different. Why would you want to do that in the first place? And now I ask you, which of those spectra is a DDA spectrum, and which of those spectra is a DIA spectrum? Anyone care to guess? I will bear with me. The very sparse one here at the top right, it's a DIA scan with an 8 MZ isolation window. And the very dense one here up top is actually a DDA scan with a 0.4 Thompson isolation window. We also have a wide window DDA scan and another DIA scan. So those you can't really decide just by looking at the spectrum whether it's DDA or DIA. And we would argue that we should analyze these spectra in the same way. Hence, we try to tell you that we would like to use a unifying approach to MSMS spectra interpretation. I mentioned that we can deconvolute chimeric spectra, so we actually tried that on a data set that was recently published, and that is sort of the new LFQ bench data set published in 2022, which has DDA acquisitions and DIA acquisitions on the same sample. And if we run those DDA and DIA files both through Chimeras and the DDA file through MaxQuant, these are the overlaps that we can see. And what is very encouraging is the overlap between MaxQuant and Chimeras is pretty high. However, when we look at Chimeras on the DDA file versus on the DIA file, we actually identify many more peptides on the DDA file than on the DIA file. However, what is very encouraging is that the quantification in MS2 here on the right, on the x-axis, that's the DIA file in MS2-based quantification. On the y-axis, it's the DDA file in MS1-based quantification. Those two methods align very nicely, suggesting that we can actually use these contributions I mentioned earlier for the quantification of peptides in MS2. However, really the interesting comparison is, DIA versus DIA. So we ran the same file through Diane and really looked at what Diane is reporting in terms of identification and quantification and what Chimeras is reporting. And what is very encouraging at this point, I think, is that the overlap between these two approaches, even though they are, they use completely different methods, is very, very high. And I think that's very good for the field, that we really do identify the same things. DIA does work. So I think that's something that we should take away from. If you take anything away from this talk, then it should be this one. The intersection is pretty high, and the quantification that Diane reports and that Chimeras reports agrees very nicely. It's not in the diagonal, so we are measuring on different scales. But there's also a substantial non-overlap between the two methods. So, and on those non-overlaps, I want to spend a couple of seconds. Because we were interested why, Diane can identify 18,000 peptides that Chimeras doesn't identify, and vice versa, why does Chimeras identify 4,000 peptides that Diane doesn't get? So what we did, and unfortunately there's not very good documentation on it, so I'm not 100% certain whether we interpret everything correctly here. We did join the library that Diane generates to the experimental output, the results, and basically quantified how many of the fragments that are annotated as included in the assay are actually used for the identification. And in the intersection, that's half as many as Chimeras, so 6. And in the outer sect, that's on average 3 fragments, so a quarter as many as Chimeras uses in the intersect. Whereas in the outer sect for Chimeras, we actually use 11 fragments on average. We dug a bit deeper into these non-overlaps, and those are actually of very low abundance. These are the distribution for Chimeras on your left and for Diane on the right. What is a bit interesting is that the distribution of peptides that only Diane can identify is shifted somewhat relative to the distribution that are shared. So here there's a bit of a non-overlap, which is not apparent on the data that we obtain with Chimeras. We are not really sure why that is. Unfortunately, I didn't have the time to and the chance really to talk to Vadim about it. But I would really, love to foster a bit of conversation here. We also dug a bit deeper into the scores that are reported by the two different approaches. And here on your left, you see the spectrum similarity distribution for Chimeras, and on your right, you see the spectrum similarity distribution as reported in the Diane output file. And again, same as before with the intensity distribution, here the spectrum similarity distribution is also shifted a bit relative to the, so for the unique peptides, it's shifted a bit relative to the ones that are shared between the two approaches. So I think that's pretty interesting data, and we really have to take a look at that and investigate this further. All of that is active research on our end, trying to move forward as a company and with Chimeras. In addition to looking at distributions only, we wanted to also check every single PSM. And within Chimeras, we have a couple of filters before we actually move all the way to percolator and FDR estimation in order to sort of weed out shaky IDs relatively early in our workflow. And when we took the PSMs that are generated by Diane and ran them through what we call our trace mode, just figuring out where we lose them, so to speak, in our workflow, we saw that 20% of those PSMs actually do not contain the base peak of our prediction. So the most abundant peak of the prediction is not present in the experimental spectrum. 20% lack the top 2 of the top 3 most abundant peaks, which I find a bit puzzling because you would expect that as the peptide elutes, it sort of the most abundant peaks come out of the grass first, which apparently is not the case for these particular peptides here. And then another 40% are weeded out by having a too little contribution to the experimental intensity, and 20% are removed by percolator. We also did an entrapment analysis at the precursor level. Again, target decoy FDR is on the x-axis, entrapment FDR on the y. And as you've seen previously with wide window data, also in DIA data, we are pretty spot on. I should mention at this point, this is a very hard case. 9-fold shuffled sequences, those are very, very similar. You will very likely not encounter this when you normally analyze your files. So this is a very synthetic type of experiment. However, in these settings, Diane seems to be a bit anti-conservative with respect to its estimation of FDR. However, unfortunately, it doesn't report decoys, so we cannot really calculate FDR ourselves. That would be, I think, a great addition in the future so that we can actually do this analysis a bit more properly. However, focusing a bit more on the commonalities and looking at quantification, this here is very encouraging. It's the most abundant peptide in the data set, quantified by Diane on the left and by Chimeras on the right. For Chimeras, this sort of XIC plot is a bit different in the sense that we on the below the x-axis, we have our prediction scaled by the contribution that I mentioned earlier, right? So at every single point, we multiply our fragment ion intensities with the contribution for that scan and trace that over retention time. And I think it's very encouraging that that those values correlate nicely with the experiment. And with that, I would like to move from DIA a bit further into another technique that uses XIC type of analyses very frequently, and that is PRM. So we hypothesized if that works nicely for DIA, why shouldn't it work for PRM? And what we did is we did a HeLa DDA run. We picked 52 peptides across the entire intensity range and did a PRM run on these peptides and then ran that analysis with Chimeras and manually extracted XICs with Skyline. Funny thing, Chimeras actually, in addition to those 47 targeted peptides, identifies 2,000 additional peptides, which is why you can actually do FDR in these types of analyses. But what is really encouraging, in my opinion, is the fact that whatever Skyline quantifies in terms of its XIC-based quantification, we can actually reproduce with Chimeras. So again, here this sort of mirrored XIC plot where the intensities correspond very nicely to one another. And if you do that more systematically and now sum up the contributions of every peptide and trace that across retention time, what you get is these Chimeras coefficient or contribution over retention time plots. And you see that in that particular window here that was used to target the orange peptide, there's actually a couple more peptides that are eluting. But what I found most striking is the fact that when we look at manual quantification using Skyline by an expert and compared that quantification to the quantification done in MS2 by Chimeras, we get the plot here on the right. Chimeras is on the x-axis, Skyline on the y-axis, and that is, really bang on the dia, a bit below, arguably, but really, really parallel to the diagonal with a very high correlation coefficient, meaning that you could, in theory, just in the future, plug your PRM data into Chimeras and don't worry about which of the fragment ions you actually use for quantification, because Chimeras can actually assign them proportionally to the peptides that you want to quantify. And with that, I'm at the end. Just a couple of takeaways. I think I've shown you that Chimeras is very promising, not only for DDA, but also for DIA and PRM data. We can do deconvolution of spectra and in a spectrum-centric fashion, and coefficient-based quant by tracing contributions over retention time. And I think I can concur with what was said yesterday in the DIA workshop, that we can predict a bright future for ML-based applications. And with that, I'd like to acknowledge the team, without which that wouldn't have been possible. Also the fantastic team from Proteome Discoverer, and I'm happy to take questions, but if you don't manage to ask them now, this is a QR code to our Discord server. Just shoot some questions over there, and we're happy to answer. And if you are one of the unicorns that does proteomics and bioinformatics, feel free to contact us. Thank you.
Thanks for the talk. I wondered if you could comment on the robustness of Chimeras for different experimental conditions. For example, varied collision energies or lower resolution MS2 scans. Sure.
So we took a look at varying gradient lengths, for example. So as you decrease the gradient length, chimericity in the spectra becomes more. So if you then analyze such data with, say, a typical approach such as sequest, and with Chimeras the gains become higher because it's just denser. On lower resolution data, Chimeras does not perform as well. And the reason for that is that the ion traps don't have as much capacity as the orbit traps. So whereas on orbit traps, we can identify in some cases, rare cases that is, but in some cases up to 11 peptides per spectrum. In ion trap that goes never beyond 5.
Thanks.
Sure.
Thank you for the very nice talk. Can you give some insight about the reason of using 8 M/Z isolation window for the DDA data?
So I wouldn't advocate for 8 at the moment. What we've seen is 4 is sort of a good compromise. And the reason for doing that is that if you increase the isolation window, you basically intentionally multiplex your scan. You intentionally co-isolate peptides, but you can later on actually separate them once again with chimeras. So you bump the numbers of IDs that way.
So a follow-up question is that in the figure you show that more than 40% increase. What is the isolation window for that data?
For the DDA data, you mean?
Yeah, yes.
I believe that was 3.8.
Cool, thank you.
Sure.
Can you provide a little bit more insight into how many fragments are shared across PSMs? Like in the example you showed, it was very nice. Only the Y1 was shared, which makes sense. But how many peaks are typically shared, or how much explained intensity? And do those PSMs then also map to new peptides and proteins, or peptides and proteins that were already identified by more PSMs with more unique peaks? For example, in protein inference, we all accept now that we should use parsimonious approaches, whereas here, in a related problem, we don't do that then.
Mm-hmm. So maybe to your first question, we didn't do an analysis sort of quantifying how many shared fragments we have across how many peptides and to which proteins they map. But what is of course clear is that fragments that are of lower mass, they have a much higher likelihood of being shared. But that's certainly an analysis that is very, very interesting to do. I should say, to your second question with respect to parsimony, protein grouping at the moment is performed by Proteome Discoverer in a parsimonious way. So that, of course, doesn't apply to the peptide approach, the identification approach here, which is technically not parsimonious in that sense.
Thank you for the talk. It was very interesting. Have you compared your ML-based approach to simpler, like top-end methods that seem to perform quite well? Is there an advantage there from your machine learning, or is that just something that's able to compress the search space so you save a little money on Amazon?
Can you maybe rephrase the part with respect to simpler top-end methods? I'm not really sure to which you allude.
So you present this kind of black box AI that's taking these highly multiplex spectra and assigning multiple peptides within them. Mm-hmm. And there are techniques out there where we search the precursor against a list of fragments, and mm-hmm. we get very good IDs without machine learning.
Sure. So one of those approaches, of course, the precursor detector is implemented in Proteome Discoverer, which duplicates the experimental spectrum for every precursor in the isolation window and then searches those spectra separately. Alternatively, you could take a spectrum, match a single peptide, then subtract the peaks and move on. But that has an inherent danger in the sense that you could either use experimental intensity multiple times, or you could use experimental intensity not often enough. So we have compared, we are outperforming sequenced with precursor detector by a margin, by a big margin. I think we should really, take good care that we don't use experimental intensity multiple times, and we do that here. And that's actually more, so we predict with AI, we predict intensities, fragment ion intensities with AI, but the deconvolution, that's linear algebra.
Let's thank Martin one more time. In the interest of time, let's move on.
Thank you.